Hey everyone, welcome to my channel Lenovate. Today we will going to discuss the structure of the hemoglobin and the types of the different hemoglobin. So here you can see I have drawn a structure of the hemoglobin in a zoom diagram that you can easily explain that how the structure of hemoglobin is made. So coming to the point that the iron that is present in the central position of the hemoglobin, this iron have 6 bonds. 4 bonds of the iron is present with the nitrogen of the porphyrin rings. These are the porphyrin rings on the 4 sides of the iron that give their nitrogen towards the center, and that nitrogen form the bond with the iron. So iron have 4 bonds with the nitrogen of the porphyrin ring, and the 2 additional bond combine with the carbon and the hydrogen, and ultimately it combines with the oxygen on the one side and histamine on the other side. Because the hemoglobin is an important carrier of oxygen, it can carry about 4 molecules of oxygen at one time. While that of the myoglobin, that is another protein of the muscles that giving the color to the muscles, carry about 1 molecule of oxygen per myoglobin molecule. Another important thing about the structure of hemoglobin is that it is a quaternary structure with a 4 polypeptide chains combined in a way to form the quaternary subunit of the hemoglobin. Here you can see the 4 polypeptide chain consisting of the alpha 1 chain, the alpha 2 chain, the beta 1 chain and the beta 2 chain combined in a manner to form the quaternary structure. So this polypeptide subunit is formed from the 2 identical dimers when combined with a bond called as the polar bond in between or the non-hydrophobic bond. When this beta chains combine with the 2 identical dimers of the alpha chain, there is another polar bond in between the 2 alpha chain, and when this beta chains combine with the alpha chain, in between these chains there is a bond called as the hydrophobic bond or the hydrophobic interactions that combine these beta chains with the alpha chain. So normal hemoglobin having a 2 alpha chains and a 2 beta chains that causing the alpha 1 beta 1 chain and the alpha 2 beta 2 chain, and combined in a manner to form the quaternary structure. And in this space there is a bond that will going to broken down when the hemoglobin is going to get the oxygen. When there is oxygenated hemoglobin, these bonds get broken in the way that in this way you can see that this alpha beta dimer 1 combined with the alpha beta dimer 2. In between there is a hydrophobic interaction that are the bonds in between, and these bonds here you can see these weak hydrophobic bonds is present in the deoxygenated state when there is no oxygen in between the atoms of the hemoglobin. Then there is a strong polar bond in between the alpha and the beta and the alpha and the beta, 2 identical dimers. But when there is a oxygenation take place, here you can see they are adding of the 4 molecules of oxygen. There is a breaking of the bonds between the alpha beta dimers. In the bonds between these two dimers, these bond got broken and the hydrophobic interaction got loosened, and there is a attachment of oxygen in between these two dimers, and that's why their carrier of the oxygen, there is a hemoglobin, carry 4 oxygens of the oxygen molecules of the hemoglobin in this way. So this state is called as the relaxed state of the hemoglobin, in which there is a loosening of the bonds between the dimers so they can carry oxygen easily. But when the deoxygenation occur, these bonds are again got strong and there is no space for the oxygen to occur in between, and there is a tightening of the bonds. That's why this stage is called as the T stage or the taut stage or the taut structure of the deoxyhemoglobin, that change into the relaxed structure when the oxygenation of hemoglobin occur. Now coming to the types of the hemoglobin we can found in a normal individual. So during the life there are different type of hemoglobin and different amount of concentration that we can find. So these are the 5 different types of hemoglobin that include the hemoglobin A, that in having the chain composition about alpha 2 and the beta 2. Here we have discussed that hemoglobin in the structure of the hemoglobin, that was the hemoglobin A or the adult hemoglobin. Two alpha chains combine with two beta chains, that's the chain composition of the hemoglobin A. And the total fraction of the hemoglobin A in the body is about 90%. So 90% of our hemoglobin is the hemoglobin A. Then the hemoglobin A2, that having the chain composition, that is the alpha 2 and the delta 2. These two chains combine with the hydrophobic interactions to form the hemoglobin A2, and the concentration of hemoglobin A2 in the adult individual is about 2 to 3%. Then the hemoglobin F. The hemoglobin F is the fetal hemoglobin because it is present mostly in the fetus. So the chain composition is about alpha 2 and the gamma 2. These gamma chains are present only in the fetus. At the adult stage, these hemoglobin is changed with the hemoglobin A, that is the adult hemoglobin. But in the fetal hemoglobin, this alpha 2 and the gamma 2 present the composition of about less than 2% in the total fraction of the hemoglobin. Then coming to the special hemoglobin, that is present only in condition of diabetes or the glycolysis condition. This is called as the hemoglobin A1C. That's a special name given to hemoglobin having the alpha 2, beta 2 containing the glucose in between. When two chains of the alpha and the beta combine with one another and the one molecule or two molecule of the glucose combine with that chains, it is called as the hemoglobin A1C or the glycolytic hemoglobin. Also it is called as the glycohemoglobin or the glycolytic hemoglobin because it contains the glucose. So the concentration is about 4 to 6% in a normal individual, but this hemoglobin increase in the diabetes or other diabetic conditions. So this was general explanation about the structure and types of the hemoglobin. I hope you will be explained well. Subscribe for more videos. Thank you for watching our video.